Okay, hopefully the nth time is the charm on my effort to get a screencast recorded on giving additional guidance and perspective on quantitative analysis of domain movements in proteins. So I'll provide a little bit of background, try to go through this quickly, but to flesh out a little bit of the context about why what domains are and why domain rotations are important. In brief, a lot of protein activities, the vast majority of meaningful biological activities, perhaps not all, but a huge fraction of them, depend on conformational changes. And a somewhat crude breakdown would be to say that there are sort of three different kinds of conformational changes. This is a bit of an oversimplification. Many conceptual models are somewhat or simplified, sometimes somewhat oversimplified, but they can still help you think about things and be highly productive in analyzing and understanding and even designing. And so a slightly oversimplified version of the landscape of protein conformational changes would be that you could divide them into three kinds of changes. So number one would be an order-disorder transition of the backbone, meaning the backbone, some segment of the backbone, or potentially even the whole domain, is in a randomly configured state, so sampling the whole Ramachandran space, and then it undergoes a transition to an ordered state where it adopts a defined conformation. So one kind of conformational change would be an either local or more global order-disorder transition. The other kind of conformational change would involve a local change in Ramachandran angles, so essentially a local conformation change within a domain, and that's definitely also an important component of contributor to protein, more complicated protein functions. But a third kind of conformational change is a rigid body rotation of the domain. And so what is the domain? And what is the domain? So Wikipedia is a mixed bag. Like all information, whether it's coming at you from Nature, the journal Nature, or Science, or Wikipedia, or the New York Times, or Fox News, all information can be of variable quality, irrespective of its source. Wikipedia is a bit more variable than Nature, but Nature is pretty damn variable. And so you have to, any information you're taking in, you have to assess, how reliable you think it is or isn't, and use your critical judgment. In this case, so Wikipedia is great in many ways, but also it can be dubious at times. So, but this entry is actually quite good. It's not exactly what I would have written about this, but I think it's actually quite well done, and to me, this. It's a very useful reference. So this is just wikipedia.org, wikiprotein_domain. And so the first sentence sort of sums it up. A protein domain is a region of the protein's polypeptide chain that is self-stabilizing and that folds independently from the rest. So the simplified definition of a domain is that it's an independently stable folding unit. That would mean in principle you could cut it out of the rest of the polypeptide chain if it's in a longer chain, and it would still adopt its native conformation with some reasonable degree of stability. And so to go on here, it says each domain forms a compact folded three-dimensional structure. Many proteins consist of several domains. Skip down here for a moment. This is sort of a typical kind of diagram you'll see. This is showing a very long animal, human protein that has many, many different domains. And so each one of these colored things is supposed to be an independently stable folding unit. So especially in higher eukaryotes, it is very common for proteins to have this kind of multi-domain structure, and regulatory proteins, especially in eukaryotes, can have many, many domains in a single polypeptide, sometimes separated by long intrinsically disordered segments. So anyway, but the domains would be the structured regions, and regions that will maintain that structure independently of the rest of the polypeptide. So one domain may appear in a variety of different proteins. Molecular evolution uses domains as building blocks, and these may be recombined in different arrangements to create proteins with different functions. In general, domains vary in length from about 50 amino acids up to 250 amino acids in length. The shortest domains, such as zinc fingers, are stabilized by metal ions or disulfide bridges. There are some domains that short that don't need that, but that is actually rather rare. The smaller domains usually do have either a disulfide bond or a zinc atom ligated by a number of side chains. A zinc atom at the core sort of binding to a number of side chains that help maintain the structure, the independently stable structure of the domain. Domains often form functional units, such as the calcium-binding EF hand domain of calmodulin. These are sort of random examples, but they are illustrative. Because they are independently stable, domains can be swapped by genetic engineering between one protein and other by chimeric proteins, and it actually has an increasing number of therapeutic applications in human medicine, something called CAR-T proteins, which is a chimeric protein that is engineered to essentially manipulate the immune system to do things like go after cancer cells. So anyway, the concept of domain is quite important in protein science. It hasn't been talked about otherwise in the lecture or in the notes, and so I thought I'd quickly cover that. So how do you find or identify a domain? The example we're about to go through in practice that is got a little bit of complexity to it. In practice, for the problem set that you're working on, the hemoglobin problem set, in that case, the definition of the domain is totally straightforward because each subunit forms one domain, and each domain is one covalent subunit of the protein. So that's an example of what is common, that one chain, polypeptide chain, is one domain. But it's also common that one polypeptide chain will contain many domains. And one example of that is shown here, pyruvate kinase. This is a critical enzyme in interior metabolism. They've helpfully colored the different domains differently. You can see that the domain, so this has 3 domains. They've colored the domains differently. You can see that this is sort of a spatially coherent and tightly packed structure that is somewhat separated from this spatially coherent and tightly packed structure. You get down here and it's a bit more complex. This is a clearly spatially coherent and tightly packed structure. And then you can see there are these little sort of add-ons to that, which have been assigned to this domain. So this sort of is an excellent example because it shows that in general, it's not, you can get a reasonable guess at what the domains are just by looking at the structure and seeing, looking for what regions are intimately associated with another rather than, more sparsely associated with one another. Down here, it shows that there are some gray areas in this, and that the domain model is somewhat of a simplification, but again, an extremely useful simplification. And then the importance of being able to calculate and quantify domain rotations or domain movements is that a hell of a lot of protein function and functionally important conformational change is involved rotations between the domains. And hemoglobin provides one example of that, and the one I'm going to go through in pymology now is going to provide another example of it. So let's go on and do that. In brief, I am going to do something, I'm not going to do exactly what's shown here, but I am going to use the same molecules. And so this is essentially covering slides 201 through 212 in the February 14, 2022 version of the slide deck. Again, I'm not going to go through everything that's here in this exact order, but the essential things that are here I'm going to capture and explain to you. To hopefully enable you to understand more clearly and implement more clearly these ideas and to apply them to the hemoglobin problem set that you're currently working on. Okay, so what we're doing here is we're looking at an enzyme. This enzyme, this is an enzyme I should say these are representatives of an enzyme superfamily. A protein family, it refers to a collection of proteins that have similar architecture and three-dimensional structure. A superfamily means an incredibly large group of proteins that all have similar structure, and usually, almost invariably, that means that they have a common evolutionary origin and they descended presumably from some common ancestor, and then by mutation the family diverged to have, in some cases, divergence just reflects speciation. You get random changes that preserve function. Well, evolution works by having random changes in sequence that then lead to, well, usually deficits, or either neutral effects or deficits in protein function, but occasionally to improvements in protein function. And then natural selection operates by selecting the random mutations that have the most benefit, while eliminating organisms containing genes that have deleterious mutations. So there is sort of just a drift component to evolution. As organisms diverge, there will be random mutations that are neutral, but also in the course of evolution, you will presumably, well, in a number of ways, but one being genetic duplications, you will get genetic duplications of a domain, of a protein coding sequence, so you'll get two copies. Just because of the way DNA replication works, well, probably DNA replication has a tendency to actually, in some cases, fail in a way where you duplicate a sequence. And that's an evolutionary advantage, because that means you can duplicate the sequence and then you can have more latitude for the sequence to evolve. And through that process of gene duplication and divergence, you can basically acquire new functions. And so in a protein family, one of the sources of variation will be just random variations between different organisms, but then also you can have mutations that change the function, but while maintaining the basic architecture and basic fold of the protein. So these proteins here are representatives of something called the amino acid deaminase superfamily, and I'll show you the chemistry of that in a moment. But basically, this performs an essential chemical step it performs essential chemistry related to synthesis and degradation of amino acids. Because there are 20 amino acids, and enzymes are usually stereochemically specific, with 20 amino acids, they're essentially, you need 20 deaminases, one for each amino acid. And so the amino acid deaminase family is represented in most organisms, probably represented to some extent in all organisms. And in many organisms, you will have up to 20 of these things, one for each of the different amino acids. So what we're going to look at here are two different amino acid deaminases from different species, but also with different amino acid substrates. So the magenta and the yellow molecules here are coming from, one of them is from a Clostridium, which is a adventitious bacterial pathogen, a significant clinical problem in the hospital setting. Often antibiotic treatment, which does essentially wipe out some of the flora, the bacterial species in your intestines, can when you take antibiotics, there is a great reduction in the bacteria, the commensal bacteria, the essential bacteria in your gut, and at that point, sometimes this Clostridium bacterium can then colonize the gut and take over and actually be pathological. So that's why there's a significant study of Clostridium. The other one, I think, is E. coli. And one of these is a glutamate dehydrogenase, and the other one is a leucine dehydrogenase. So these are from different species that are pretty widely diverged, and they actually have a different amino acid substrate. But you can see that they're very, very similar. And what we want to do is dig down and look more carefully and quantitatively at exactly how they're the same and how they're different. And so that's what we're going to do now. Before we do that, I'm going to quickly go over to look at just the chemistry quickly. Actually, the overall chemistry that these things catalyze is shown here. So glutamate is, of course, one of the 20 canonical amino acids. Alpha-ketoglutarate is one of the intermediates in the TCA or Krebs cycle. So these are both extremely important metabolites. It turns out in many organisms, including E. coli, glutamate is the most abundant free amino acid in the cell under almost all growth conditions. It's actually used for a couple different purposes in addition to a precursor for protein biosynthesis. In any event, to get from glutamate to alpha-ketoglutarate, you need to remove this amino group. That is a two-step reaction where first you actually remove H minus here, and when you then take the hydrogen and 2 electrons away here, the lone pair electron, well, this is the protonated form of the primary amine. The proton will be removed. The free electron pair will then make a double bond with the carbon when you remove this hydride group over here, and so you'll get something called an imino group to produce iminoglutarate. And then this thing essentially will hydrolyze, meaning the NH2 linkage, sorry, the CN double bond is considerably higher in energy than the CO double bond. So in a water environment, water will rapidly replace this. But it turns out that in the case of the catalyzed deamidation of amino acids, which is done by the enzymes we're about to look at, these two steps both occur on the enzyme. The first step involves the donation of the hydride group to something called NAD, which is a major energy carrier in the cell. And so just to remind you, NAD is nicotine adenine dinucleotide. This is just AMP basically here. Then there's a second phosphate group into the ribose. That's why it's a dinucleotide. And then rather than one of the 4 canonical bases, it has this nicotinamide base. And this is a critical cofactor because this will accept the hydride, an H minus, a proton with 2 electrons on it. And so NAD and NADH, which NAD plus is shown over here, NADH is shown over here. They differ simply by the addition or removal of the hydride. And so this is a major energy source in cells and an important substrate for an innumerable number of metabolic reactions that involve hydride transfer. And so the enzyme that we're looking at has to bind NAD plus and the amino acid in a spatially precise way, and then do some chemistry on it. And what we're going to see is that a important component of that, the structural mechanism that helps mediate that catalysis, is that there is a domain rotation. This, I think, is a leucine. I forget. This is either a leucine or a glutamate. Anyway, it's the amino acid substrate that is bound to in one of these structures. You can see it is located between these two different sort of halves of the molecule. And what we'll see shortly is that basically this is one domain down here, and this is a second domain up here. In the catalytic site where the amino acid gets deaminated is at the interface between the domains. A key part of the mechanism here is that the domains either swing further apart or clamp down to either allow the substrates and products in and out, right? When the domains swing out, the substrates and the products exchange more efficiently, and then the domain swings in and clamps down and actually usually dehydrates the active site. And in that dehydrated, closed conformation of the active site, the catalysis takes place and the chemical conversion. And so this kind of domain movement is very common. Catalytic sites are often, in fact, usually located at domain-domain interfaces. And the rotation of the domain often plays a key role in, again, letting the substrate in, letting it out, or clamping down to produce a structurally well-defined catalytic environment. It's often also an important point that is then isolated from bulk water. So the domain rotation around the interfacial catalytic site is a significant contributor to the function of many enzymes. So we're going to then, that's why this is sort of an interesting enzyme to look at and a good model for looking at domain movements. Okay, so now we're going to jump ahead and do that. And so there are 2 of these amino acid dehydrogenases that we're going to compare, again, from different species and with different amino acid specificities, but still you can see they are remarkably similar in terms of their overall tertiary structure and fold. So we're going to load these 2 things. We'll get 1 BG feed first. Okay, that's 1 molecule. That's very clear. We can click on that, and it's going to tell us that it is glutamate. So this is the glutamate dehydrogenase. This is the small molecule in there is what we had looked at in the previous slide at the domain-domain interface. So one of the things that we're going to do in this case is when we select the domains to start doing domain alignment, we're actually going to try to keep that amino acid with in the internal representation primal of the domain. Okay, so that's one of the ones we're going to be looking at. And the second one we're looking at is going to be 3SBO. Okay, now this looks more complicated. And what's going on here? Well, as we spoke about in class, the unique region in the crystal, which is always a regularly repeating three-dimensional lattice containing many, many copies of the same molecular assembly, the asymmetric unit of the crystal is the unique part in terms of the atomic content. And when we solve a crystal structure, we solve just the asymmetric unit, and that's what you'll get when you open a crystal structure file from the PDB. So this is the asymmetric unit of this enzyme. You can see there it's much bigger, and that's because you have the same subunit repeated here multiple times. And so it turns out that this is a hexameric assembly. I think you can sort of see, if I run it exactly right, you can see that there is that there's sort of a threefold symmetry axis here. Well, let's move on. It's in any event. But anyway, this thing has sixfold symmetry, and actually, this is like hemoglobin. This is a cooperative enzyme, and so as I told you, glutamate is a important metabolite in addition to being an amino acid precursor, and so glutamate metabolism is very, very regulated. And it turns out that in the same way that the oxygen binding affinity of hemoglobin changes when the subunit interactions change, the catalytic competency of this changes when these subunit interactions change. And this is actually another, like hemoglobin, this is actually another classic example that has been used to study the molecular basis of cooperativity. So we're not going to do that today, but we're going to focus on the simpler question of how the domains in the individual protomers are moving around. Okay, so let's, we want to just compare a single subunit of this. And so to do that, I'm going to break this down into its constituent subunits. As opposed to the structure we looked at before, there is no ligand bound to this. They do have a ion here. This is a chloride ion. That is probably, well, it's present in the crystal structure. When you see something like that, it is debatable what its physiological importance is. It could just be there's unusually high chloride in this solution just to make it, to crystallize it. But in any event, that's not one of the substrates. And so it could be, that probably is a preferred chloride binding site. May not be very important. It's certainly not a substrate for this reaction, so we're going to ignore that as we go forward. So the first thing I'm going to do is take this thing and break it into its constituent subunits. So we're looking at them one at a time. To do that, I'm going to go to chain selection mode, and I'm actually going to do this from the sequence bar because that will enable me to get the letters right for the subunits, right? So subunits in the PDB are necessarily labeled by letters, and it starts with capital A, then goes capital B, etc., etc. Okay, so I can select this either in the sequence bar, or once I know which one I'm looking at, I can select it down here. I can verify that's domain A. I'm going to copy that to an object, and I'm going to rename that object 3SBO. This is 3SBO-A. And now I'm going to try to find B. And I'm going to copy that to B. And rename it. And I'm going to then go to C. Copy it to another object. Rename it. And copy, go to D. It's a bit tedious. Copy to D. Rename it. And we're almost going to get we're almost at the interesting part of this. But anyway, we do also want to show you all the steps so you understand exactly what's going on. Again, I'm going to rename this to E. Oops, there was a mistake. Let's sorry, rename objects. To verify I didn't screw that up, I'm going to turn off everything for a moment, just make sure that is really A, which it is. Good. So far, so good. Now I'm going to go back to F. This should be F. Oops, not there. This should be F. Yeah, it's because the what's this projection? I grabbed the wrong one. Yes, this should be F, which it is. Copy to the object. Name object. Now, in the hemoglobin exercise, you wouldn't have to do exactly this. You sort of have the opposite problem. You've got to build up the native tetramer of one of the two one of the two variants, conformational states of hemoglobin. And again, we're only going to, in the end, focus on one subunit of this enzyme. But one of the tricks in doing these analyses is just keeping everything organized. And two helpful things. One is to use clear, simple names. Number one, and number two, this could be a good hint for the hemoglobin exercise. And number two is to use color to distinguish things in an easily detectable way. Okay, so I'm going to delete the selection here. And again, what that does, it's confusing to me when it's up here in the menu by deleting. And when I delete one of these molecules, they just disappear from memory, as I mentioned before. If I delete the selection, it just resets that group of the selection is the active group of atoms. It just wipes out the active group of atoms. So it really does have quite a different effect in practice than deleting one of the objects that's in memory. Okay, so now we have all four of these. We have separated these out to 6 constituent subunits, and so now I'm going to go back to this structure. I will center it, and now I'm going to get the domain. And so we're getting onto the interesting, whatever the relevant part for hemoglobin. And now, where is the domain? Well, in hemoglobin, again, it's going to be quite easy because each subunit is a domain, and that's like, and each domain is a subunit, so that's easy enough. In this case, it's more like we were looking at the wiki entry. We have to have the spatially coherent, sort of self-interacting part of it. And you can see that this is, looks very intimately associated and spatially coherent. This looks very intimately spatially coherent. There are 2 helices at the interface here that are, are a bit more ambiguous in terms of which of these domains they would be in. And again, the domain model is to some extent an approximation, although an extremely useful one. And so in this case, there is sort of an ambiguity whether 1 or 2 of the helices here, which domain they belong to. But in any event, I have worked that out so that you don't have to worry about it. And like up here, right, this is very clearly a coherent spatial unit. And so we're going to look and see where that is. So you can see that's at residue. Oops, chain. Sorry, I'm going to now go to residue selection. And I am going to just check, select just to see where this is. This is at 233, and I'm going to then just see what's down here. And so that's residue 448. But if you look at where that helix is actually sort of coming sort of down here to the C-terminus, and you can see that before that, up to this point, right, so this C-terminus comes back, goes over here, blah, 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 blah. So let's just try to narrow down to, a segment of this that is just, looks very clearly to be self-contained and self-interacting. And so I'd say that would actually be about over here, right? Like here, this would be, oops. Again, there is a slightly tricky point about defining one of the domains. Again, with the hemoglobin example, you're not going to have to worry about this. Okay, so we could, like, this beta sheet is clearly up in this domain, right? And so let's go see where that is. And so that's at 368. And then what I'm going to do is just going to track that sort of to the end of the molecule. And that is all roughly in the upper part. And then when you get to the end here, though, it extends down. Now, when we get to the very C terminus, we get a region of this domain that's sort of all the way down here, and now associated with this lower domain, right? So again, in this case, the polypeptide chain actually starts out at the, well, let's go back over there. Again, this is a slight complexity, but one that you will not have to deal with in the hemoglobin exercise. Here's the N terminus of this thing. If we start from the N terminus and go on, right, this is all clearly the spatially coherent unit at the lower half of the screen, spatially coherent unit at the lower half of the screen, spatially coherent unit at the lower half of the screen. This is all like pretty damn clear cut. And now we're clearly have gone across some boundary and we're going into the other half of the molecule, right? So as a first guess, I'm going to say, well, that's one domain. And if I look at the C terminus of the molecule, right, I can see that like this helix is intimately associated with this stuff we just selected. And if you look in here, this way the cells project, right? This helix is where we looked at the C terminus before. This is intimately associated with this N-terminal domain. And so it does look like this is technically we call it sort of a domain swapping. So there's a C-terminal helix that is part of the this folded domain. And that is somewhat of a, that happens, and that is not uncommon. It is somewhat of a complexity for the domain model, but again, the domain model is still extremely useful. In this case, you can just say the domain is constructed by somewhat remote regions of the polypeptide chain, which is a bit of a pain in terms of selecting it and isolating it, but this is still a spatially coherent folding unit. Okay, and so now we're going to just take the part of this that's in, just crudely speaking, in that domain. This is largely, right, N-terminal material. This is the N-terminus plus a tiny bit from the C-terminus. And before we saw that glutamate substrate is actually, I think, mostly associated, right? There's the glutamate, and it's sort of more associated with this. So it is at the interface between the two domains, but it is sort of more in this domain. So I'm also going to take the glutamate. And then the first thing I'm going to do, having selected this N-terminal domain in its extension in the substrate, I'm going to color this differently, and so in this case I'm going to make this a blue. And so now I've got the N-terminal domain sort of labeled, as it were, in blue. And you can see that the rest of this is mostly a very clearly coherent, spatially self-associated unit. What's going on down here is a bit more ambiguous, but to keep things simple for today, we're just going to call everything that's in this region, including this single helix, we're going to call this all one domain. And we'll see some interesting things about that in a moment. And so those are offsetting colors. We can just stay with that. That's all good. But now what I want to do, for a number of reasons, I want to create a separate object for the N-terminal domain. So that's now going to be quite easy because I've labeled it in blue. I can find it again very easily. I'm going to oops. That was, yeah. And sorry, that should be blue too. That was just, that accidentally got omitted. That is in this domain. Right, so let's go again. So I'm going to go here, and I can use shift click to get the entirety of the N-terminal part. I just got to grab the C-terminal extension here in the glutamate, and now I've selected the entirety of this domain and one of the two substrates for the enzyme. I'm going to copy that to an object new, and so I'm going to call this 1BGV enter for N-terminal domain. For N-terminal domain. And now you can see I can turn off the whole thing, and I will, right, so now I have just the N-terminal domain and the glutamate, which is now in the same color. Maybe I'll color that slightly differently, make it a little bit easier just to keep track of it. Ooh, sorry, bad color. But now I can just color the whole object, and this selection here is now just this one. I'm going to color just the selection. Cyan. Okay, so that looks good. And now I'm going to make a copy of this, I hope it's lying, because that would be like the nth calamity for trying to get the screencast completed. But I'm going to ignore that for the moment. Let me see how serious that is. No, there's a gig available. I don't think that's not serious at the moment. Well, to be safe, the download folder has a huge number of cryo-EM maps. That's why this is not such a full disk, really. Actually, that small problem there for my other work. Yeah, these guys I went back. The rest of them I'm going to empty the trash. Empty trash. And this is going to free out. That's weird, but I'm not going to worry about that. Yeah, so this will make sure our disk is stable for the rest of the screencast. Okay, so back over here. Now, I just want to make another copy that's just the C-terminal domain. You'll see in a bit why that's going to be a useful thing to do. And I'm just going to copy this. There, I'm going to delete selection just because I get confused when it's sort of floating in the menu up here. I'm going to rename this to, guess what, 1BGV C-term for the C-terminal domain. And now you can see that I have cleanly separated this thing into two domains, N-terminal and C-terminal. And so now I could, for instance, take one of these subunits. This is just, we'll zoom out. I could take one of these subunits and do an overall alignment. Let me start with that. 1BGV-A. Oops, sorry, 3BSO. 3. Yes, being slightly dyslexic can be a small impediment to doing this. And now I'm going to align this. To begin with, I'm just going to align it to all of 1BGV, and we'll take a quick look at that, which again is a single chain. Actually, if I just go one, it'll autocomplete. And now I'm going to let's do this alignment, see what happens. So that white, the A subunit there will align. And you can see that's actually pretty good. And you can see that these proteins only have about 50% sequence identity, so it means about half the amino acids are different. And you can see there's three-dimensional structures are very, very similar, and the domain organization is equivalent. And so this isn't a bad alignment, but you can already see that the alignment is somewhat better down here in the blue region than it is in the peach region. And so what I'm going to do now, so already we're seeing some evidence of a difference in the domain alignment, alignment between the two domains here. And then I'm going to get that, I'm going to clarify that essentially by, instead of aligning that subunit to the whole of B1BGV, I'm going to align it just to the lower N-terminal blue domain. And so that's the enter. It's very convenient that Python has auto-completion. If you type a string of something that's in memory and you hit a tab, it will either autocomplete or it will give you a list. If I go there and I just hit tab, it gives me, it says, oh, there are two objects that match the parser, a C-term, N-term. If I type N, I don't have to type ter because I can autocomplete. Okay, so now I'm going to do the alignment rather than the whole thing, and then watch what happens here. Oh, this alignment got better. This one is now more off, and you can see that there is quite a distinct rotation between these domains. You can check to see whether the domain structure is conserved. Instead of aligning with the N-ter, I can align with the C-ter. Up. That's really beautifully aligned, right? There's a little bit of deviation, but not much, right? That's actually beautifully aligned. So this domain structure is highly conserved between these significantly diverged homologs. And now I can go back to the N-terminal alignment, and this structure is preserved. And so what this already is like a very important message, right? You can see that the main conformational difference between these two proteins is in a rotation of the domain. So this would be called a rigid body, a mostly rigid body rotation of the domains. That means the domains are mostly rigid. They are maintaining their folded structure with very little change. But there can be some local changes. Those can be important functionally. But the main thing that's happened here is that this domain has, as a single rigid unit, rotated relative to this domain. And of course, that rotation has happened around the catalytic site, which is at the interface. So now what I'm going to do is I'm going to actually align all these other subunits. So I'm going to turn them all on. I'm going to zoom out. So you can see the whole damn thing. I'm now going to just one by one align everything in 3D. All the subunits in procedure. So bang, aligned. Bang, aligned. Again, just to the N-terms, bang, aligned. Bang, aligned. and we should be done. Okay, and now I'm going to zoom in on everything because we got a little set of them, and I'm going to turn up the sequence bar so we can see for a minute. And so what do we have going on here? Well, the domain concept, again, is helpful and illuminating to us because we can see, wow, that's pretty, all 8 of them are pretty damn similar there. But if we look up here, what do we see? Oh, there's a slightly different orientation of the second domain. I won't do the exercise just to save time, but if I did the C-terminal alignment of all of these things, we would see the, well, let me do that, it's not so long. Let's just do the C-terminal alignment. There's F to the C-terminus, E to the C-terminus, D to the C-terminus, C to the C-terminus, B to the C-terminus, A to the C-terminus, and guess what? Man, those are pretty damn conserved too, right? And so, whereas now here things are in an array of slightly different positions, and so this shows there's essentially a flexible interaction between these 2 domains, and in the different copies in that E. coli structure, you get slightly different angles between the N-terminal domain and the C-terminal domain and all the different subunits. So that already tells you, again, you can see very clearly these are pretty rigid units, but they're rotating to one another around the active site at their mutual interface. Okay, so let's go back to the N-terminal alignment and get to business, and we are getting close to the calculating the rotation angle. We're almost there. There's a tiny bit of additional setup to do. And okay, so these N-terminal domains are all, like, again, beautifully similar. And now we can see, oh, okay, wow, there are a bunch of different sort of rotations of that domain. And let's, I think we're going to make this the easiest exercise to follow if we find if we focus on the domain that has the single largest rotation. So let's figure out what that is. Okay, so if you look here, oh, there's a beta sheet, right? These 2 beta sheets are very similar in structure. This helix is very similar in structure, but if we, this is obviously a large rotation, and so I think if we look at this amino acid, well, I think this one is like, this is essentially, just by looking, you can tell this one has rotated the most. What is that subunit? That is subunit F. Right, it does look like subunit F has the greatest rotation. Let's just take a look at, right? Oh yeah, that's a pretty big rotation. We can quickly compare the others one by one. That's a lesser rotation, although that one is very similar, but it is a lesser rotation. That was how it's E is very similar. D is clearly less, C is clearly less, B is clearly less, A is clearly less. Actually, the analysis of the allosteric regulation of this enzyme, again, it's a whole field of study. You can find many papers on it. It would be sort of interesting. But that's not our topic today. Our topic today is just to quantify, understand domains, quantify, and understand how to, the operational procedure for characterizing domain rotation. And so really what we want to do now is just quantify this rotation. And we are, at this point, almost ready to do that. And so we're going to focus for the rest of the screencast on just the most rotated domain and calculating the rotation angle there. But it is important to note, right, that the different subunits all have different rotations. So basically, in the state of the E. coli enzyme, these things are sort of free to rotate around relative to one another to a significant extent, and in the different subunits in the E. coli enzyme, they are trapped in different conformations because the crystal lattice will only form when these things are in a precisely defined spatial geometry. You have to have consistency to better than 1 atatomic diameter between the unit cells over the course of 10 unit cells to get interpretable strong enough diffraction to solve the structure. And so you need a very, very high degree of spatial organization. What has happened is the lattice is constraining these individual subunits in specific packing angles. But in the different subunits, you can see there are different packing angles, and that means that in solution, this thing is just probably wobbling around, exploring all these different packing angles between the 2 domains that surround the catalytic site at their interface. So I'm not going to show you this today, but it turns out that This is not the fully closed structure. There are other homologs in the protein database that have fully closed structures. It turns out there's about another 10-degree rotation here to actually fully close this active site, and there are other structures in the PDB that have that. But to keep it somewhat simpler for today, we're now just going to focus on this domain rotation between these two C-terminal domains. So to do that, I'm actually going to bring back the sequence bar, and I am going to now look at the, I'm going to color the C-terminal domain differently. And so like I can see there that there's actually the end of the blue thing. And so it turns out that that's at residue 204. And so we're going to go there in 3SBO. Oops, something is... Yeah, no, everything's good. Yeah, so I'm going to go there at 204 in this subunit of the E. coli enzyme, and now I am going to just grab the whole domain, and I'm just going to color it differently. Oh, I see what's going on there. Yeah, I'm going to color this whole domain differently, so I'm just going to trace out the entirety of the page domain. And so that'll be everything except a few amino acids at the C-terminus, right? Like over here, we down right over here. This is what we have defined as the peach domain. That's roughly correct. And so now I am going to color this whole thing yellow because it will stand out from the rest. Okay, so now in the right set looks like you got it about right. Okay, so in the 1BGV, the Clostridium enzyme, the N-terminal domain is blue. In the C-terminal domain is peach. In the E. coli enzyme, the N-terminal domain is white. Those are beautifully superimposed. The C-terminal domain is yellow. So what we want to do now is calculate the angle between the yellow and the peach domain. And to make that just a little bit clearer, what I'm going to do is I'm just going to make a copy of the yellow domain. And so I'm going to copy it to an object, new, and I'm going to, again, delete that because I get confused when it's not at the bottom of the list. I'm going to rename this object, right? This is 3SBO subunit F, and this is the C-terminus of that. Okay, so now I can just turn off the N-terminal domain from the Clostridium enzyme, and I can turn off our model of the full, right? And now we have just the C-terminal domain of E. coli in yellow and the C-terminal domain of Clostridium in peach. And so all we want to do is calculate the rotation angle and axis in this script written by a member of the Pymol user community. Is then available, which he is distributing freely for everyone who uses Pymol to use. We can say we can do this calculation. So I'm going to the source is by convention of Pymol the first model. So I'm going to say I want to calculate, and of course the rotation is symmetrical, but we're going to say let's go from the C-terminus of Clostridium, so from 1BGV C-ter, and again we need quotation marks because this is written in Python, and Python requires that on strings. Oops, it's got to be 1BGV, and I'm going to calculate that to 3 BSO C-term. Sorry, I want to go from 3SBO. Sorry, I want to go from 3SBO-F C-ter. Yes, exactly, to 1BGV C-ter. And looks correct. Let's let it rip. Okay, and so it's thinking. And now what has it done? Okay, so it's brought up a couple of new objects. And let's actually hide our initial 1GVZ C-ter. Sorry. Let's hide our initial, 3SBO C-terminus. And now you can see, right, that if we look at our original alignment, oops, sorry, clicking the wrong buttons. sorry, 3S3, yeah. If we look at our original alignment, let's get it working for the moment, right? Like these things are obviously displaced. But after what it has done is it has calculated the proper rotation matrix to superimpose the yellow, as well as can be done by something called the least squares minimization algorithm. It has, found the rotation angle that minimizes the distance difference between the backbone atoms. And you can see now, if we compare these two yellow molecules, right, there has been a rotation of the two yellow molecules. Actually, let's change the color of one of them just slightly. Let's make this a little bit, maybe that's perhaps too subtle. Yeah, it's still a bit subtle. This is going to... yeah, so you can see that that these are clearly have moved, right? And if we look at, right, so this is the same, to be clear, the working is the rotated version of the one that we put in, right? The working will be the rotation version of 3BSO-FTER, right? And so yeah, so these are the same domain just having been rotated. And if we look at the working version, we will see that it is very well aligned, right? So now I brought up, this is the, what I did is I looked at the, right, the yellow is the, the yellow is the C terminus of the E. coli enzyme subunit F. And if we compare that to the C terminus of the Clostridium enzyme, right, they are not lining up well, but that's because there has been a rotation. And once you find the optimal transformation matrix relating them, you can rotate that into a position. Oops, sorry. You can rotate that into a position where it. Sorry. Yeah. So now what we're looking at is we're looking at the rigid body rotated version of the C terminus of the E. coli subunit F in the C terminus of the Clostridium enzyme, and you can see that is once again a beautiful alignment. And if we bring up the other half of the Clostridium enzyme, and then we bring up the entirety of the E. coli enzyme, sorry, of the E. coli subunit F, right? Like you can see that even though these are only about 50% identical, there's just a beautiful alignment of the entire assembly. But when we look at it in the crystal structure, the C terminus of the C terminus of the C terminus of the F subunit, the E. coli enzyme, has a substantial rotation. But when we use a mathematical algorithm to figure out how to rotate it and optimize the superposition, we see that there is quite a this is the yellow is the original position of the C terminus of subunit S. The beige is the rotated position of the C terminus F. If we, so it's the same molecule just rotated. If we look at the alignment of this to the C terminus of the E. coli enzyme, that looks very far off. But we take the rotated version of the same thing, right? So this is the non-rotated. Now we take the rotated, and bang, it is a beautiful match. And so this has shown us that, again, we concluded this before. We did individual C-terminal and N-terminal superpositions. We saw the domains are quite conserved, and they're really just moving around as rigid bodies. Now we have actually calculated the transformation matrix that takes the starting position of that and figures out exactly how you rotate it to get it to align up perfectly with the Clostridium enzyme. And basically this axis shows the rotation axis. And so it's now depicted in three-dimensional space. So you can see this is the axis around it with its rotating. And here, actually this number, I'll turn these things off for a moment. The number here is actually the rotation angle. And there's a theorem in multivariable calculus that says if you have two things that are related by some kind of rigid body transformation in three-dimensional space, you can define that uniquely by a rotation around a certain axis and a translation parallel to that axis. And so all of those numbers are actually given to you up here, as well as the description of the direction of the axis. So this describes the orientation of the axis. This says the rotation angle is 13.5 degrees. And this says the length of the translation vector along rotation axis is 0.51 angstroms. So that means this isn't a pure rotation, but what it means is, and we can then look down the barrel of the axis. This is sort of an interesting exercise. We look down the barrel of this axis, and we take our starting location, and we take our ending location, and I'll turn off the sequence thing for a moment, and you can see that, oops, that's a rotation. Oops, where did our axis go? Oh, I'm sorry, axis is right there, but we can't point it out on the screen at us. Right, this is a 13.5 degree angle rotation, and so this is a overwhelming, this is overwhelmingly a rigid entity. There are relatively few conformational changes within the domain. The entirety of the domain as a rigid body rotates 13.5 degrees along this axis coming out of the plane of the board. And it is also translating, but it's only a 0.5 angstrom translation, which is very low. It's actually the distance between those things is actually 0.5. So you can see that this is overwhelmingly a pure rotate. This is almost a pure rotation. There's a very small translation involved in it. And now we can bring this up in the context of the entirety of the structure that we started with. And so again, the cream is the rotated position. The yellow is the starting position. We can bring back the N-terminus of 3 SBO by doing that. Right, so what we see here, this is all 3 SBO. The white is the 3 SBO N-terminus. The yellow is the starting position of the 3 SBO N-terminus. The cream is the rotation. Right, sorry, is the rotated version that superimposes with the C-terminus of the other enzyme. And so what this has told us is that the main conformational difference between the Clostridium enzyme and subunit F of the E. coli enzyme is a 13.5 degree rigid body rotation of this domain. This is going in the direction of clamping down on the the glutamate substrate is there. This is moving the enzyme domains closer to encapsulating the substrate, but it hasn't gotten all the way there. There are structures of other homologs in the PDB where this thing rotates down around another 10 degrees and completely closes the active site. In practice, you only see that when the NAD substrate is also present in the enzyme. But so there is a more closed form of it, but it involves similarly a rigid body rotation, a larger rigid body rotation to clamp down these domains. But in this case, this gives a very, I think, clear and nice example of all of the concepts and in practice how you do this. So again, a big point here would be that what we're seeing here is this is now right. So the axis now is coming out of the plane, the screen towards us. We can look at it this way, right? And so again, the rotation now would be sort of into the plane of the board. There's the rotation looked at that way. You can turn it around here. Here's the rotation that would come out at us, right? And if we look right down the axis, we can see that this is almost, there's a small translational component, but this is almost a pure rotation. And if I'm going to bring back 3BG just for a moment. So I'm going to make a separate copy of this for a second. And I'm going to just show spheres for this. Right, so that's the glutamate substrate, and you can see that our rotation is moving this closer to encapsulating the glutamate substrate. And if we have NAD, when the enzyme actually turns over, that would involve NAD plus binding here, and at that point, this rotation would be even greater in magnitude. But we can see that between this snapshot in the of the E. coli F subunit, this F subunit snapshot of the E. coli amino acid glutamate deaminase, and the Clostridium enzyme, there is an 11 degree rigid body rotation. And this I think, illustrates how well that you can have very significant domain rotations. In this case, the domain rotation is of critical functional importance, because what you're seeing is that without the substrate's bound effects, actually this thing is sort of wobbling around. The crystal lattice sort of constrains that wobbling interaction into a number of discrete states. Right, in the crystal, we have a number of sort of discrete orientations, but those are just stabilized by the crystal packing. In solution, this thing is just wobbling around, sort of waiting for the substrate to arrive. When the substrate arrives, it rotates down and clamps down over here. In the meantime, we can see that the F subunit in that crystal lattice is rotated about 13 degrees compared to the conformation you see in the Clostridium enzyme. And this shows you how to calculate domain rotations, and hopefully also informs you about what a domain is, and gives you more insight and perspective on why they're important.